Welcome to Chapter 3. Let's go ahead and get started. Okie dokie. This presentation won't talk about absolutely everything, but I'm going to give you a good tour of some of the things you need to know, okay? Starting with this idea of chirality, okay? If something is chiral, it means that it's a mirror image. So your hands are a mirror image if you hold them up like you see in the picture here. But I cannot take, for example, this hand and bring it over and superimpose it on that hand and make it identical. Notice that this thumb here would be on the other side. Okay, you can prove that to yourself. And all this is saying is that when you have a central carbon atom surrounded by 4 different groups, you're going to have this, it's called chirality, okay, which are the mirror images, and it makes them what we call stereoisomers, and the molecules are different with different chemical properties. Okay, this next image here is talking about the idea of polymers. And as we know, polymers are components that are composed of monomers or single units. So if you are dealing with, for example, carbohydrates, you are going to have monomers of monosaccharides that form polymers, and this could be starch or cellulose or whatever, and they're all linked together. Okay, in DNA, you have the monomer units, which is called a nucleotide, and we'll talk more about that later, and they form polymers here, which would be DNA or RNA. So many nucleotides to form DNA and RNA. Proteins, you have amino acid as the monomer, many of them in long chains form the proteins. Okay, proteins are the polymers. And of course here in the lipids, you're going to have the fatty acid, which is basically a big long chain of carbons and hydrogens, and many of them together would form the triglyceride. So just the idea of monomer and polymer. Okay, the macromolecules, and what do they do? Okay, so this is a great little table, and subunit basically means these are the monomer units, what makes them up. Function, it's great. What are they used for? And here's a great example. So what examples would I want you to know? So in the carbohydrates, I like the idea of, for example, cellulose versus starch and glycogen. If you look at these two here, you see what these two polymers do. Okay, they're polymers of glucose, but they're very different. All right, so understanding that starch and glycogen is for energy storage, and cellulose, which is also glucose, though, as we talked about in class, is for structural components. Nucleic acids, we'll get more to that later. Proteins, for example, you can have structural proteins like hair and silk, things like keratin and silk that are polymers of amino acids, and you can have things like the hemoglobin here, which are functional proteins and will actually carry oxygen in the blood. Okay, and lipids, you have the phospholipids, prostaglandins, steroids, terpenes, fats, etc. There's a lot of different lipids depending on where they are in the body. Okay, just some examples there. Okay, this is very important in biology, a dehydration reaction, dehydration synthesis. If I take any two monomers over here, okay, so monomer unit here, and I remove water, that is how I form a bond between them, okay? And conversely, if I add water, like here on the other side, I can break the bond back into the two monomer units. So dehydration synthesis and hydrolysis, okay? This is very important. Oftentimes you'll see, instead of reaction, you'll see it dehydration synthesis. Synthesis means to make, to synthesize. Okay, so you either are removing water to make it or adding water to break it. Okay, what do we have here? We have here the idea of the 5 and 6 carbon sugars. Okay, ribose and deoxyribose. These sugars are used in the nucleic acids, DNA and RNA. And then these 6 carbon sugars are the 3 simple sugars, the monomers, and glucose is usually the one we're going to be talking about. Glucose is blood sugar. It's the one that's primarily used. It's present in starch and cellulose and etc. You do see fructose, fruit sugar, The FR is for fruit, remember? I'll just put fruit. And galactose is simply a stereoisomer. Remember, the hands superimposed a stereoisomer of glucose. Okay, so just an idea of where we find these sugars. So these are monomers. This is in the book. You can take a look at it. Alpha glucose and beta glucose. So I will draw attention because this is the difference between starch and cellulose. Notice over here where this hydroxyl group is relative to each one of these. Everything else is the same except for these hydroxyl groups. One's up and one's down. That's that chirality. This carbon here is a chiral center. It's got the 4 different groups as we talked about with the hands, and by moving one of the groups up or down, we don't change the formula. The formula is still the same, C6H12O6, but we get a different structural isomer, and whereas alpha glucose on the top will lead to starch, beta glucose will lead to cellulose. So it's amazing what a small change in the stereoisomer can make a huge difference in the molecule of the polymer. Okay, what do we have here? Again, this is just going over glucose and galactose stereoisomers, whereas fructose and glucose are structural isomers. Notice there's actually a structural difference, whereas the only difference between glucose and galactose is the position of that hydroxide group. So just different ways to have different isomers, or same chemical formula, different structure. All right, here is your using the monomers, okay, mono, using dehydration synthesis to form disaccharides, okay, such as sucrose and maltose. All right, just to give you an idea of how that happens. And these are great for carrying sugars around the body. Okay, what is this? This is an example of, you have your nitrogenous base phosphate group, and you have your sugar. This is a nucleic acid. Or I should say, this particular is a monomer of a nucleic acid, okay? And nucleic acids are made of nucleotides, okay? So you have this here, this nucleotide, and nucleotides are formed of a nitrogenous base, there are 4, a coordinating sugar, and a phosphate group. And it's going to be different in DNA and in RNA, okay? And notice the difference you see down here. This little square box will determine, well, if it's a hydrogen, it's in DNA. If it's a hydroxide, it's RNA. Okay, and that's really what makes the difference. Otherwise, the rest of this is pretty much the same. Okay, so nucleic acids are made of nucleotides. Okay, and this is what we look at. You see several nucleotides. Okay, so we have nucleotide, 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 and they all form together in these polymers. All right, these are your purine bases. Okay, adenine and guanine, and these are your pyrimidine bases, cytosine and thymine. And in RNA, you get uracil, and uracil replaces thymine. Okay, so if you're RNA, you'll have uracil instead of thymine, okay? And these are the 4 bases that write our code, our A's, T's, C's, and G's. Remember the movie Gattaca? G-A-T, oops, T-A-C-A. That was what that was referring to. Guanine, adenine, thymine, thymine, adenine, cytosine, adenine, okay? If you were actually to read the title of the movie Gattaca in DNAEs, okay? All right, and here's how we link them together. Your DNA is a double helix, and you're going to have these bases that are going to be hydrogen bonded together. Hydrogen bond is like a weak bond, but you get millions of them, and it's pretty strong. It's like Velcro. Okay, so hydrogen bonds are like Velcro, okay, and they hold DNA together. And we know that DNA, okay, is the molecule that stores our information. That's our genetic code, so it's very important to protect this information, which is why we Velcro it and wrap it up in a double helix. Okay, RNA is a little different. RNA is single-stranded. As we see, there's no other strand. okay, whereas DNA is double. And that's for protection primarily. And RNA is more expendable, so it's single-stranded, okay? And we have the same bases except for uracil in place of thymine, okay, and deoxyribose sugar instead of ribose sugar. Okay, well, deoxyribose in DNA, ribose sugar in RNA. Okay, this is just ATP. Notice the difference, or the similarity. You've got a nitrogenous base, in this case it's adenine. You've got a sugar, and you've got 3 phosphate groups. Okay, adenosine triphosphate. Okay, so nucleic acids can be used for our, and this is our energy molecule. I'm going to put it as a big E. It's our big energy molecule. Okay, and here just is a great table talking about the many functions of proteins. Okay, so we're going to pause there for this particular phase.